Hello, my name is Mecca Wallace-Spurgin. This is an example of an absorb type activity. Today we're going to spend some time talking about cellular structure and function of a eukaryotic cell. Here I have an example of an animal cell as well as a plant cell. And I've already gone ahead and circled our nucleus in both the animal cell and plant cell, our mitochondria in both, along with our Golgi apparatus. Those are three organelles that we will talk about today. Our goal is at the end of our PowerPoint presentation, you will have learned several organelles as well as their functions. So these are three examples of organelles that you will be learning about. The first area of our cell, our eukaryotic cell that we're going to talk about, is cytoplasm and cytoskeleton. Next, talking about cytoplasm and cytoskeleton, the cytoskeleton is the supportive structure within the cytoplasm. Scientists used to think that the cytoplasm was simply a fluid-like area that organelles just randomly floated around in, and that's not the case. Actually, you can see right here, mitochondria, endoplasmic reticulum, ribosomes, those organelles are actually attached or embedded within the cytoskeleton. And the cytoskeleton is made up of two substructures, one called microtubules, and these are thin hollow-like tubes that are made up of proteins called tubulins, and then we have our microfilaments, and those are very hollow, thin fibers as well. Our nucleus, we have here our nuclear envelope, and you can see I drew a circle, kind of a circle around the nuclear envelope. It's also known as the nuclear membrane. In the nuclear envelope, there are small tiny holes called nuclear pores, and this allows for larger substances to move into and out of the nucleus. And then we have in the center a nucleolus, and we're going to talk about the nucleolus more in detail here momentarily. So inside of the nucleus, the nucleus is responsible for directing all the cell's processes, and it contains DNA. And as we said in the previous cell, the nucleus is surrounded by what's called a nuclear envelope or nuclear membrane. And in that membrane, there are tiny holes called nuclear pores. And like we talked about, larger substances can move in and into and out of the nucleus through those holes. Ribosomes. Within the nucleus, there's a nucleolus, and inside of that nucleolus is where proteins are manufactured. Now, ribosomes are a little different from other organelles. They are not bound by membranes. And the site of ribosome production, as I said, takes place here in the nucleolus, but they don't stay there. Instead, they are released, and ribosomes can be found, you can see them circled here, freely outside in the cytoplasm, or they are also found here in the rough endoplasmic reticulum. Talking about our endoplasmic reticulum, it's also called ER, and the rough ER ribosomes are attached, and you can see these little bumpy circles here that I have circled in red, and these are our ribosomes. And so ribosomes found in the rough ER are actually attached, and in here they produce proteins that's used for export to other portions or other parts of the cell. The smooth endoplasmic reticulum here that I have circled in red, notice that there are no ribosomes attached. The smooth ER is responsible for the synthesis of carbohydrates and lipids. Smooth ER is also found in your liver, and it helps to detoxify harmful substances. The Golgi apparatus. The Golgi apparatus is this flattened stack of membranes, and its job is to modify, sort, and package those proteins from the ribosomes into these little sacs called vesicles. And these vesicles then, notice that this one is a newly formed vesicle leaving the Golgi apparatus, and what happens is they release proteins in these little vesicle sacs into the environment inside of the cell, and they travel then to the plasma membrane oftentimes, and that is where the proteins are released into the environment outside of the cell or outside of the plasma membrane. Vacuoles. Here in our plant cell, we'll take a look here at plant cells. We have this very large vacuole, and what this vacuole does is it stores things such as food and enzymes and other materials needed by the cell, such as water. Some vacuoles also store waste products. Now I want you to take a look here at our animal cell, and you're not going to see a vacuole typically in an animal cell, and if you do, they're much more smaller in size than in plant cells. Lysomes. Lysomes have an additional name, and they are oftentimes called a suicide sac. Their job is they're the cleanup crew, so they digest excess or worn out organelles and other food particles. They also digest bacteria and viruses. The membrane, they are covered by this membrane on the outside of them, and inside of there we have these digestive enzymes, and because of that membrane, the digestive juices are not allowed to destroy the lysomes or other parts of the cell. Lysomes will also at times fuse with vacuoles. Remember, we said that some vacuoles store waste, and so the lysomes then will digest those wastes. And here is just a cross-section of our plant cell, and notice that we have a nucleus here in the center, and lysomes are typically located, here's another one, very close to that nucleus. Centrioles. Centrioles are made up of groups of microtubules. They're often found or always found in pairs. They're located in the cytoplasm, again near the nucleus. And centrioles are necessary for cellular division, which we will talk more about in chapter 9. And then we come to our mitochondria or mitochondrion in singular version. And so mitochondrion, they have this outer membrane here that you can see that's kind of this brownish color. And then on the inside, they have a very large surface area, these little folded sacs of the inner membrane here. And mitochondria are important to the cell as they are kind of the energy generators. They convert fuel particles, mainly sugars, into usable energy for the cell. And so the energy produced from breaking down of those sugars is stored in bonds of other molecules and used later on by the cell. And so that's why oftentimes we call the mitochondria the powerhouses of the cell. Chloroplasts. Now, chloroplasts are only found in plant cells, and inside of chloroplasts we have these small flattened disc-like shapes that are considered to be thylakoids. And we know that plants receive their energy from the sun, and so within those thylakoids, this is where the energy from the sunlight is trapped inside of these thylakoids, and then of course through the process of photosynthesis, we're able to convert that light energy into chemical energy that the plant can use. Plants also have a cell wall. Animal cells do not. So out here, this green is the cell wall, and it actually surrounds the yellowish part here, which would be our plasma membrane. So cell walls are thick, rigid mesh of fibers. As I said, they surround the outside of the plasma membrane. They protect the cell wall protects the plant cell and gives the plant its support, and this cell wall is made of a carbohydrate known as cellulose. Cilia and flagella. And so the two main differences between cilia and flagella, cilia, notice they have lots and lots of little hair-like projections, and they move in the manner of oars, like you're rowing a boat. And then you have an example of flagella, and these projections move in a whip-like motion. They are typically longer and less in numbers than cilia, as you can see, most often just one or two. An example of a cell that would have a flagella would be a sperm, and so we see just one long projection there, and again it moves in a whip-like motion. In summary, we've summarized several structures of eukaryotic cells, both of plant and animal cells. And one of the main characteristics that distinguish plants from animals is that plant cells contain chloroplasts, and they can capture the energy from the sun, as well as plant cells have cell walls that provide protection for plant cells and support for those plants. Another way that make animal and plant cells different is plant cells typically have a large vacuole and animal cells do not. If they do, the vacuole will be very small in shape. The next slide, we'll just talk a little bit about sort of summing this up. Cell structures work together to perform cell functions such as protein synthesis, energy transformation, digestion of food, excretion of waste, and cell division, all which help us and other living organisms maintain homeostasis, of course. This last page here I want to point out to you as your glossary. We will follow this PowerPoint up with a 12-point little quiz covering the structures as well as the functions. I encourage you to go back and visit the glossary to help you be successful in the quiz and upcoming assignments that you have. Thank you.